Hello, and welcome to the Editing a Workflow video module, which is a part of the 7 Bridges onboarding lesson. While watching this video, please keep in mind these key concepts. The first is that the public apps gallery contains hundreds of apps that can be used in many types of scientific analysis. These apps are parameterized and can be easily modified with our workflow editor. Workflows can be reverted to previous versions when needed. At the end of the video, you will be able to navigate through the public apps gallery, modify and save changes to a workflow, and finally, revert a workflow back to a previous version. The apps gallery is curated and contains the tools you need for analysis. There are over 800 tools and workflows. The benefits of using these apps is that they have been optimized for speed and cost in the cloud. They are fully customizable and can be run using the graphical user interface or via an API client available in Python or R. If you click on any of the cards you see in the picture to your right, you'll find a helpful description for each tool and workflow. All of the tools in the apps gallery are reproducible, portable, and pre-configured. This is made possible by describing each piece of software in the Common Workflow Language, also known as CWL. CWL is a community-based open standard designed to make scientific workflows more portable and reproducible. Each step in the workflow is enabled by Docker, which provides all the necessary software dependencies for that step to execute. Our online editor gives you the option to just click and drag to add tools to your workflow. Double-click on any of the individual tools to customize them for your needs. Not only can you easily build a workflow with our editor, but you can also access previous versions. This way, you can go back and run an analysis you did several iterations ago with the same exact settings and parameters. In the platform demonstration, we will do the following. First, we'll navigate to the public apps gallery and we'll copy a workflow to your project. From there, we will edit the workflow, and finally, you'll see how to revert to a previous version of the workflow. In the browser, I have navigated to cgc.sbgenomics.com. From here, I've created a new project space for this demo. Just to get you reacquainted, on the left you'll see a description space. We can modify this by clicking Add Description. On the right-hand side, you'll see a list of the members in my project. Currently, it's just me. And then any analysis that we run, you'll see on the bottom right-hand side. So the first thing that we want to do is navigate to the public apps gallery. So I'll click on Public Apps, and then Workflows and Tools. So it says currently we have 862 different apps. I'm going to click on this button to browse all of our apps. So I am going to search for just a typical workflow, maybe RNA-seq. And from here, we'll copy a few of these over to our project space, and I'll show you how to modify these workflows. So if you scroll down, you'll see several different options. We see here a high-sat string tie workflow. I'm going to copy this to our newly created project. So it's going to onboarding video, editing a workflow. Copying that to our workspace. And then let's do another one, maybe this Callisto salmon sleuth workflow. So if you click on any of these workflows, you'll be taken to a new page where you see a description of the workflow. You can click Read All to get additional information, some common use cases, modifications introduced by 7 Bridges, and some important notes. You also see how to implement this workflow using an API call in Python. And finally, you'll see some benchmarking information, how long it takes to run approximately, depending on the file sizes, and how much that will cost. At the very bottom, you'll see the different ports, so different types of inputs, some app settings, and then the expected outputs for this workflow. So what I'm going to do is just copy this over to our project space. So I selected copy, and I'm moving it into the onboarding video project space. Okay, so it says an app has been copied to your project. I can click on this and it will take me back to our project space. Notice there are several different tabs here. The dashboard page. Files is where I can add my data or find publicly hosted data. And apps is where these new workflows have been transferred. I'm going to click on the HiSat string tie workflow. Again, you'll be given some basic information about this workflow and how it runs, and I want to show you how to edit this workflow. So I will move to the top right corner of the screen and click on Edit. A warning will come up just letting you know that you are about to edit this workflow. So click on Edit, and then you'll see what this workflow looks like. So basically we're going from reads to HiSat for alignment, and then string tie, and then eventually you'll be given some DE-seq transcript count matrices and gene abundance estimations, things like that. So just to make a basic change, we can easily search for a tool and click and drag it into this workflow. So I see that there is no quality control here. The reads go directly into pair FastQs by metadata and then directly into HiSat. So if I were to introduce something like a FastQC, all I have to do is search for FastQC in the Public Apps tab, and I can click and drag one of these tools into my workflow. So FastQC is requiring an input file, so if I click on this node, you'll see that the reads light up in green here. So I can directly connect the FastQC input to the reads. In addition to requiring an input file, we need to also click on these output nodes to get a zipped report as well as an HTML report. I'm going to zoom out in just a sec here. So if this is looking a little bit messy, what you can do is click on Auto Arrange on the very bottom right corner here, and it will kind of organize all of the apps so that it's easier to follow. So now the reads are not only going to pair FastQs by metadata, but they're going directly into FastQC before then moving on to HiSat and string tie. Any of these can be modified as well by double-clicking. And you'll get some app info is the first tab here. And you'll be able to modify any of these input nodes, change app parameters. I can specify the number of CPUs we should run it with, things like that. And then you can also change the ID name. So the label here is what appears in our workflow. I'll just change it to FastQC, and you'll see that instantly changes. So once you're ready, you can click Save. And I'd like to add a revision note, so added FastQC to the workflow, and click Save. So for our next example, we have a Callisto Salmon Sleuth workflow. If you click on this, again, you'll be taken to a new page with some information. So we start essentially with a list of FastQ files and an index file. Then we go through a tool called Choose Pseudo Aligner. So this gives the user an option to either use Salmon or Callisto, and from there it will go to Sleuth for differential expression. I'm going to show you how to edit this workflow. Let's say that you want to use Callisto and Sleuth together, but you want to modify this so that when a user uses Salmon for quantification, it will directly go to DE-seq. So let's edit this workflow. Again, you'll see a warning pop up just saying that you're going to edit the workflow. And again, I'll navigate to the left side here and click Public Apps and search for DE-seq. So from here, what I'm going to do is click on the tool that I desire, which in this case is DE-seq, and drag it over to my workflow. Okay. So you'll see that there is one input node and 5 different outputs. So for the input, if we click on this and pull it out just a little bit, we see several different options. I can attach it to salmon's transcript level quantifications or gene level quantifications. In this case, I'm going to just choose transcript level quantifications. I also have the option to attach it to Callisto if I'd like. In this case, I'm going to leave it just attached to salmon. From here, we see several output nodes. So we have analysis results, an R data file, a phenotype file that comes out, normalized counts, as well as an HTML report. Okay, so now we have our reads going from salmon to DE-seq. You'll notice that they are still attached to sleuth. If I'd like, I can just click on one of these attachments here, and then go to the bottom right side of the screen and click the trash can, which means delete. Okay, and so now what we have is a workflow where we have FASTQ files, and then you choose the pseudo-aligner. If a user chooses salmon, it's going to go directly to DE-seq2. If the user chooses Callisto, it will then go on to sleuth. And for either of these differential expression choices, you'll get a list of our data files, analysis results, normalized counts, and an HTML report. And again, if this is looking a little bit messy, you can click on the Auto Arrange button on the bottom right-hand corner, and it will kind of organize your workflow a little bit. So from the beginning, we have a list of FASTQ files. The user will choose which pseudo-aligner they want, and it will either go to salmon and then DE-seq, or it will go from here to Callisto and then sleuth, and either way you'll get a list of different output files, normalized counts, HTML reports, and so forth. Once we're ready, we can click on Save. So I like to add a revision note. Okay, once you're ready, click Save. And sometimes it takes just a second to save. You'll see the page has refreshed, and we can click the name of the workflow to go back to that page. And you'll see now DE-seq is added to this workflow. I've showed you how to modify 2 different workflows. Once we're actually ready to run the workflow, we simply go over to the right-hand corner of the screen and click Run. You'll notice that we're running revision 1 of this app. If you'd like to run a previous version, let's say the high-sat string tie workflow before we modified it, right next to Run, you'll see the revision options. We can go to revision 0, which is the original version. You'll notice in this one, there is no FastQC. So it goes directly from reads to pair to high-sat. Again, revision 1, we've added FastQC here. So it's on the very bottom. So it goes reads to FastQC. But if you'd like to run an older version, just click the original revision, and there is no FastQC here. Again, we can click Run, and it will create a new task with this older version, revision 0 of the workflow. If at any point you're stuck, confused, or have questions, you can click on this question mark button on the bottom and describe your issue. I hope you now have an understanding of how to modify a workflow. Some key takeaways to keep in mind are 1, again, the public apps gallery contains hundreds of apps and workflows for many different types of scientific analysis. I showed you just a few today. 2, these apps can be easily modified to fit your needs with our workflow editor. And 3, you can easily run previous versions of the workflow. Our next video in the module list is building an app or tool.